Everyone, I'm Jack Cush with Room Now. I'm joined by good friend, Dr. David Liew. Dave, let's introduce ourselves. Jack Cush, Dallas, Texas.
David Liew, Melbourne, Australia. And we've both been in Copenhagen together.
That's right. We met in the middle, so to speak. Although somehow I think his travel is going to be better than mine going back home. But we are here on the final day of EULAR, Saturday, day 4. An odd day. With really the only novel new research being presented was the late breakers. There were a number of educational sessions that were good, but we're going to talk about the late breaking abstracts. That's usually the latest, the greatest, the thing that just gets in under the wire, the latest releases from the company doing their proudest of bestest of data. So we're going to present what we think are a good selection of that. And David, why don't you start off?
Yeah, I love the late breaking session. It's always my favorite at ACR EULAR. It's a lot of fun. And I think it helps that there's a little bit of chaos because I think, people have scrambled to get the data together. So it's maybe not always as polished as a big plenaries, but there's a lot of fun. And I think deucravacitinib is something which is going to be very interesting. It was a lot of fun to listen to today. So this is the Paisley study. In lupus, that's presented by Eric Morand, my compatriot, who is up first on the late breaking stage. So Paisley is a phase 2 study looking at deucravacitinib. That's a TYK2 inhibitor, a not JAK inhibitor, or JAK inhibitor, depending on who you talk to. And that's a phase 2 study. The study design is is almost getting a little bit consistent now, but essentially it's add-on therapy to existing therapy in patients with moderate to severe lupus with a standardized steroid weaning protocol, and then you see what happens. So we saw today really good results. Good results over the overall SRI-4, as you'd expect, was the primary endpoint. Good outcomes there. When we looked at the kind of high-level things, LL-DAS, which is always a talk now, that lupus low disease activity state, we need low disease activity as well as low steroids. Those were all good as well, definitely favored deucravacitinib over placebo. We saw skin and joint data as well. It was good on those. We saw some serological data in the form of double-stranded DNA and C4, and we saw those substantially better in the deucravacitinib arm versus the placebo arm. Now, we didn't see, of course, other clinical data, and I think perhaps one of the things that's always been bandied around about some other lupus therapies is that maybe they're stronger on the skin and joints, and there's a lot of argument about the other lupus involvement. Having said that, that's an evolving space, and we've seen some pleasing evidence about that for other therapies. We would like to see what deucravacitinib looks like, and of course, I think we're very likely to see a phase 3 from the sponsor, as I hope we should, as I think we should. So, really tempting thing. Watch this space on both of those counts, the other lupus manifestations, as well as the phase 3, the next little bit. Very interesting.
So certainly exciting because it's a new molecule, although it's in the JAK class. There might be some advantages of a TYK over or JAK inhibition, and people have spent a lot of time looking at the safety of it. Are you seeing those JAK-related serious events we're concerned about, like Mason cancer and zoster, and so far, and they're not really seeing any of that. But it's early in development. But I think that there are several issues around this that are really quite important. One, this worked, but this worked in a phase 2 lupus trial. I can count for you on many fingers the number of great data in phase 2 that didn't look quite so great in phase 3. Ustekinumab, baricitinib, et cetera. And again, as you saw, we talked about earlier in the week, baricitinib had its 2 big phase 3 trials, Brave 1, Brave 2, where it failed. It didn't do so well. One of the reasons was a very high placebo response. And some think that, well, this drug did so well, this new TYK inhibitor, deucravacitinib, did well because the placebo response was 34%, and the drug response at the probable dose of, I think it's 6 milligrams, it was 58%. So a good enough delta effect that that makes people happy. So I think that placebo responses are a problem in lupus trials. They tend to catch up with you around the time you do big numbers in phase 3. But this looked good. We've seen it look good in psoriasis. It beat apremilast in psoriasis head to head. It looked pretty good in PSA. So is its future going to be in the psoriatic arena, or is it going to be in maybe in something like lupus? When we asked Moran, the author who presented this, I asked the question about, this was really good joint data and skin data. Classy response rates were 16% versus 70% on the drug, 16% being placebo, and then the joint count 50. I'm not sure what that is. I assume like a 50% improvement was 45% placebo, kind of high, but a 68%, 6 milligram dose. Those all look good. We asked, where's the rest of the data? And just like you said, David, it's early, people are scrambling. He said, we haven't had time to look at all the data. And I'm sure they looked at other lupus outcomes. I'm sure they didn't include people with lupus nephritis in the trial, or lupus cerebritis, or bad news lupus. But this was really encouraging data, which is what you want to see in your phase 2. The question is, if you're a betting man, Dr. Liew, are you putting money on phase 3 or not?
Well, I think, like you say, the very promising phase through failed phase 3 is a cliche in lupus. So if I put money every time, I'd be much poorer than I am right now. So I think that's it's really hard to say. I hope for the best, and then maybe
we'll do.
See what happens.
Yeah. All right. So the next one was presented by Ian McInnes. That first one was late breaker number 4. This is late breaker number 1, LB0001, presented by Ian McInnes. It's bimekizumab in biologic DMARD naive PSA patients, a 24-week study called Be Optimal. It's a large phase 3 study. As you know, bimekizumab, it's got a lot of studies here that were being presented. It's both an IL-17A and IL-17F inhibitor, and that might be its distinguishing feature. It looks really good in skin. Head-to-head studies with other IL-17 inhibitors, the dual inhibitor, if you will, actually has fared better. The question is what's going to do in PSA, and they've got studies in biologic experience, biologic naive, biologic wannabes. I don't know, but they've got like 3 studies that are out. No, they actually got a SPA study that they presented, I guess. But the data looked encouraging. It was, as you can imagine, a placebo-controlled trial. It was placebo versus bimekizumab versus adalimumab as an active comparator. With the majority of patients, it's sort of like a 1-to-1 randomization, most patients being on bimekizumab. A good number of them, half of them were on background methotrexate going in. They had active disease, like, 17 tender and 9 swollen joints. I mean, they really had a lot going on. And at the end of week 16, the primary endpoint was obviously bimekizumab did well. The placebo response, 10%. You like to see that in clinical trials. The BKZ, that's its call letters, I guess, 44%, and adalimumab, 46%. No difference between adalimumab and bimekizumab at an ACR 50 response. And that's, kudos to them for having that aggressive primary endpoint. The other primary endpoint here was a PASI 90, another aggressive endpoint. And the placebo response there was 3%. The BKZ was 61%, and the adalimumab was 41%. So clearly, the 17 inhibitor was better than the TNF inhibitor, and that would have been expected a priori going in. There, the rest of the outcomes are kind of what you'd expect. Good things happening. As far as the secondary endpoints and the time to response and all that, I'm not going to get into. The safety of this is where things look really, really good as well. And they also had an X-ray outcome, and basically showed really no change in X-ray outcome. But then again, you'd expect that also to be the case for the adalimumab group. The placebo patients did show a very small increase. The adverse events, nothing surprising here. Very little in the way of, like 2% or something like that of oral candidiasis, and that's an issue. With this dual inhibitor in the psoriasis studies, that number, which is usually single-digit low in the IL-17 studies, with the dual studies in psoriasis, it's been as high as 15%. In the studies that they presented at this meeting for bimekizumab, that's been low. It's been around 3% and whatnot. So again, very positive trial. The very interesting thing about this is the ACR 50 here, as I said, was 44%, compared to the other bimekizumab trial, the B complete in TNF-IR patients, it had a 43% response. That was the last slide that he showed. So encouraging data, and of course they had dactylitis and enthesitis data also looked very good. David, what do you think of this study?
Yeah, and I mean, I think we've been waiting for this for some time. I know that's we've had a lot of new psoriatic arthritis drugs, and you could say that about a lot of different drugs. But I remember going back to EULAR 2018, seeing work from Dennis McGonigal's group, translational work, looking at what happens if you blocked A, F, or both, and really seeing quite a substantive advantage in translational data of dual blockade. And I think we've been watching, I think in psoriatic arthritis, we've got used to taking cues from our dermatology colleagues, and it's been a lot easier for them. They've had a lot, in some ways, they've had head-to-head studies and where there's been clear differentiations on the endpoints they really care about, relevant to the patients that they see. And so they've got excited about this. Of course, in that head-to-head of bimekizumab to secukinumab, the B-Radiant study, there was that signal with oral candidiasis, but it was manageable from that point of view. We didn't see worse in terms of incident colitis, which I think is something we still worry about with IL-17 inhibitors. So I think that we've been this has been building up to it, and the derms are getting excited about this. The question is what this means for the things that we care about, which are probably patients with lower PASIs, but with more joint disease. We haven't seen much in terms of joints between secukinumab and a TNF inhibitor. So I think, what would we necessarily expect this to be an advance on a TNF inhibitor? We'll have to wait and see. And hence the rise of the head-to-head studies, I think, is going to be really interesting.
Yeah, it will be, clearly the head-to-head studies have made a major impact in dermatology, less of an impact in rheumatology, but maybe it's because we haven't seen enough of them or the right one. So it'll be interesting to see if that comes to pass. Before we move on from this area, I think there was a study of bimekizumab in spondylitis that looked really, really well. And there was also a JAK inhibitor, upadacitinib study in spondylitis that looked really well. So it's an exciting time for drug development in this arena. So and there was a lot of good stuff in PSA and SPA at this meeting. David, let me do a real quickie here on baricitinib in JIA. As you know, baricitinib is approved for use in rheumatoid arthritis, and they went down the route of trying to develop this in kids with juvenile arthritis, and they did so in a 220 patient phase 3 multi-center study, and they did it in the typical, or maybe it's the regulatory way. They do a PK study to look at the equivalent dose, and basically the people in the study receive the equivalent dose of, I think it was a 4 milligram equivalent dose, and it's going to be weight-based dosing in kids. And they enrolled kids, 220 of them, ages 2 to 18, with polyarticular, but also extended oligo, also ERAs, also some juvenile PSA. So I don't know exactly who's in this group, but that becomes important when I talk to you about the flare rates and how many people do well on taking nothing. But the JIA design is everybody gets drug as an open label run-in, and then the responders stay in the study and go through a double-blinded withdrawal. So at the end of week 16, the ACR 30, the primary endpoint here was 76%. And that one of those 163 patients entered into the double-blind withdrawal phase of the study, which clearly favored those who are on baricitinib, where they had a 17% chance of flare off of drug, or on drug, I'm sorry, and the placebos off of drug had a 51% chance. But while that's clearly differentiating and enough for regulatory approval, the question is, why is it these kids are only half of them are flaring on placebo, followed for another 6 months or more. And why are 17% flaring on drug? And the question is, I think the question lies, maybe the answer maybe lies in the fact that there are different types of JI in this trial. If they were all polyarticulars, I think we'd see much different results. But since it's got a mixture of stuff in there, which is common in the JIA trials, we're seeing these kind of results.
And I mean, I think the thing is, I don't know much about JIA, but I do know that it's certainly not just a straight swap for our adult diseases. So I think that it's one of those things that, firstly, it's good to see that it does actually do what we would expect it to do, but it's so hard, I think, for us as adult rheumatologists to really appreciate what's a breakthrough in that space. I mean, especially, I guess, an oral agent in needle phobic kids. I think that's plausibly something, having extra options there is certainly something that they'll appreciate.
Exactly, and that is, the driving force, because while tofacitinib went through this recently, prior to that, it was a bunch of biologics that were injectable therapies, and so there is a big call for an oral agent for kids who have one of the forms of JIA. Our last one is the so-called, is it called Sapphire?
Sapphire? I think it depends on where you come from. Yeah.
It's spelled oddly, Saphyr. And what does it mean, Dr. Liew?
Well, I can tell you, I don't know what sapphire means, but I think I can tell you what the study is, which is sarilumab in relapsing PMR. It's not often that you see PMR up on the podium. Certainly not a phase 3 clinical trial, let alone a positive phase 3 clinical trial. So I think this was really interesting. I'm really keen to see the details, but let me tell you what the headlines are. So the first thing I'll say is, let me tell you, it's sarilumab 2 weekly, 200 milligrams 2 weekly with a 14-week prednisolone taper versus placebo with a 52-week taper. So in essence, they've gone with a shorter pred taper, gambled hard on that. It's a shorter taper than what we saw in, for example, Giacto in giant cell arteritis. So I think it was reasonably aggressive to start with there. The first problem they ran into was the fact that they decided to run this in the middle, largely in the middle of COVID. And so they actually ended the trial having under-recruited, only actually recruited 118 of the 280 they'd planned. But despite that, they hit the primary, they hit multiple secondaries. Now, I think that there's, all of them were treading in the right direction. Even the patient-reported outcomes, the ones that didn't quite make it, were basically on the edge. And, how much you believe in 0.05 or not, I'll leave up to you. But really, when you talk about the things that matter, sustained remission was the primary, and we saw a really big difference there. I think it's probably better to do it when you take away the CRP, and then we saw 31 to compared to 13% there. And if you looked right across the board, that's the efficacy side of things. But then you also look, on the other hand, that, as you'd expect, there was actually reduced steroid exposure, they tried to assess the stereotoxicity with the GTI, the glucocorticoid toxicity index, benefits there as well. So I think that's all really quite impressive. I think there'll be questions about exactly how things go. And I think in these kind of studies, you've got to be careful because obviously the CRP is sometimes involved in some of those, some of the endpoints. I'm sure that these investigators have done this very well. I have confidence in that, but we of course want to see to make sure that all these things have been done in a way that meant that the blinding actually stayed, rather that this is really truly double blinded. But we have a therapy that works in PMR now, and plausibly this is a basis of registration for a drug apart from PMR. We saw, just as a side point, one of my colleagues from the UK, Matt States, had data, primary care data today on the poster floor. Basically, 18% of all people which is kind of half the UK in this, 18% of the people on prednisolone were on it for PMR. And that this is something that affects 2% of the population over 55. This is not an uncommon thing. So. Right. Really, and these are patients with a whole lot of comorbidities where they can't really afford to be loading on the steroid. So I think these kind of things lining up together does give you a sense that this is something, with all the provisos that you have after seeing a late breaking session and the slight chaos that comes out of that, it's a really, really interesting space. And
so
yeah,
I'm going to give you a bunch of statements and you're going to tell me good news, bad news, or no news at all.
Yes.
This trial was done between October 2018 and I think July or August of 2020 during COVID, yet there was no COVID outcomes.
Yes, it was alluded to a little bit. I think it was a lot of patients wouldn't have been exposed to it. I think 2020 in the United Kingdom, I wasn't there, but it did sound crazy. So I would give them a whole pass on that. But I think that it's a tough one, and I think it's going to be, we're going to demand these COVID outcomes from a whole lot of studies that are occurring right now and going forward, because I don't think COVID's going away. We're stuck with it.
At the end of the trial, those that were on the placebo got a total of 2,000 milligrams of steroid, and those that were on the sarilumab active taper there was 700 milligrams. What say you?
Well, like I said before, so people with PMR have an average of 3.5 comorbidities, a mean of 3.5 comorbidities. I mean, in that UK data that we saw today. These are comorbidities that suffer when you have steroids. Certainly that kind of difference really does make a big difference. And that's not even accommodating things like osteoporosis, sarcopenia. These are the kind of things which people potentially, which not only impairs their quality of life and their functionality, but kills them when you start to get older. So I think that that's a big meaningful difference in terms of steroid. I think that these are that magnitude of difference is if it was in giant cell arteritis, you'd say that's an impressive difference as well. So I think I think that we haven't seen the same kind of data from TOSI yet, from tocilizumab. We saw PMR spare, much smaller study, not really I don't think anyone expects that to be the basis of registration. So I think this is really still on the march in that respect.
My last concern is, first off, the preamble here is that going into this trial, these patients had about a year of PMR disease activity, disease duration. So the primary endpoint at week 12, unfortunately, had a placebo response of 38%, and the active drug had a drug response at week 12 of 47%. So that's about a 9% therapeutic effect difference. But the primary endpoint was sustained remission, and that meant that you were in remission from that week 12 to the end of the year. So for the 40 weeks more, and there the numbers were 10% placebo and 26% with a delta effect of 18%. Do these numbers encourage you or discourage you?
Yeah, and I think these are really interesting questions. We obviously want to see the detail from the paper, but I think when we were thinking about PMR spare and trying to interpret that, the tocilizumab in PMR small study that Kristen Diaco ran, I think that we know that patients with PMR take a bit of time to flare. And so that early stage, you're not going to necessarily see that same effect. We don't have the details of what doses of pred they started on and how it weaned, but really it often just takes that little bit longer to really start to separate the winners from the non-winners as far as these therapies go in a disease like PMR. I want to see the details there.
All right, David, thanks so much for taking the time to do this daily recap. I want to encourage all of you out there to follow David on Twitter. He was a star at this meeting. I love that little serial tweet you did about the telemedicine debate that had its own humorous moments, and he kind of covers that in a video and his tweets. It's definitely worth looking at, folks. So safe travels home, David.
Thanks, Jack. Always a pleasure to be working with you.